Good evening, everybody. I welcome you all for the today's Ask Me Anything series session. I'm Sumitra from HTIC MedTech Incubator hosting the session. Before we move to the session, before we move to the session, I would like to say a few guidelines. I request all the participants to kindly stay on mute and please unmute by yourself when you want to speak anything or you want to ask question. We also created a feedback form for this session and we will be posting the feedback form link in the chat box. So requesting the participants to please fill the feedback form at the end of the session. So this will help us to improve our upcoming Ask Me Anything series session. So for for today's session, the for today's session we have the speaker is Dr. Bhuvaneshwar sir. So Dr. Bhuvaneshwar has made outstanding contributions in the development of implanted and disposable medical devices. He also headed Sree Chitra Tirunal Institute for Medical Science and Technology at Trivandrum for 36 years. He also played an important role in the growth of biomedical engineering and medical industry in India. He all He's also a distinguished alumnus in IIT Madras and also adjacent visiting faculty in IIT Madras. He is currently independent consultant medical devices and development and testing. And the topic for the today's question is ask me anything on types of clinical validation or investigation. So, we are happy to have you, Doctor, here for the today's session. So, usually in our AMA sessions, we will ask the speaker to set the tone for this session and then we will go forward with the questions from the participants. So over to you, Doctor, to set the tone for the session.
Thank you, Sumitra. Good afternoon, everybody. It's a pleasure to be here and to have so many participants for this discussion. So what I have planned is sort of an introduction to what is clinical investigation, or more commonly called clinical trials of medical devices. A little bit about, I can discuss how it is different from drug trials, if anybody is interested. But medical devices trials can be a little more challenging than the well-standardized drug trial formats. So what I planned is about 20-25 minutes introduction to how to do or plan a clinical investigation for a medical device, and I have one case study related to the artificial heart valve in which I've been involved for a long time, and then we can go on to the questions. So Mitra, if anybody wants in between a clarification, is that
I can let you know, Doctor.
Okay.
So once if you're done with the presentation, then we can go forward with the questions from the participants, Doctor.
Okay. Somebody wants to raise a hand. I don't know if that feature is here.
No, Doctor, the raising hand feature is not there. I can let you, Doctor.
Okay, they can then type in the question and you can.
Yes, Doctor, I can.
Want a clarification on any slide. So that's the format, and actually it's more, I'm not just talking about types of clinical investigation, it's more introduction to clinical investigations of medical devices. So let me share my screen and start my presentation. Yeah, can you see my screen and hear me clearly?
Yes, Doctor. Yes, Doctor.
Okay,
but we can't see the presentation screen.
Just a minute, I just want to mute my video. Yeah, so that. Can you see my presentation now?
Yes, Doctor. Yes, Doctor.
Yeah. So primarily, as I said, this is an introduction to what is involved in a clinical investigation. There are a number of terms. Basically, they're also called user validation or device validation. I would say clinical investigation is a part of user validation or device validation, but there could be other aspects of validation more than clinical investigation. Clinical investigation is one very important part of final validation of a device. So the primary purpose of a clinical investigation or even validation is to show that your device is safe and it performs as you claim it to be. So if you're a developer or a manufacturer, it is your responsibility to demonstrate that the medical device that you've developed is safe and will perform as you claim. So that's the underlying principle why we need to do all this. Just a background on regulatory terminology. Many of you may have heard the terms clinical evaluation. Then we have our medical device rules published by the Health Ministry. That's the MDR 2017. Many of you might have heard of it or seen parts of it. They use the term clinical investigation. MDR also uses the term clinical performance evaluation for in vitro diagnostics, which is different from clinical investigation. And the main aim is to look at the three terms which can be confusing but mean slightly different. One is performance, efficacy of a device, and the effectiveness of the device. It's also used in drugs, the effectiveness of a drug. So clinical evaluation is, do I have anything more? Yeah. So what is the difference between clinical evaluation and clinical investigation? So internationally today, these two mean entirely different things. Clinical evaluation, in fact, sort of includes clinical investigations when necessary. But you can look at the terms in the ISO standard 14155. So clinical evaluation today is internationally used. Particularly, it has become essential under the European regulatory requirements as well as the US FDA requirements. Is the review of the relevant scientific clinical literature and or the review and assessment of data collected through clinical investigation. So today, these regulatory agencies, particularly for moderate to high and high-risk devices, they require you to have a document which is called a clinical evaluation report, where you review all the relevant scientific literature for the device, as also your predicates or whatever treatment, what is the gold standard treatment for whatever you're trying to find a solution for. So a detailed review and documentation of that is required, including the risks, a good risk assessment, and what are the failures or complications that can occur. And those have to be part of the clinical evaluation report. Then comes your own clinical data of how you prove that your device will perform as you claim it to be and is safe, and the risks involved have been mitigated or controlled and are at an acceptable level. So this has become a very important term, and it in effect encompasses clinical investigation, but it is much more than that. It is partly doing something like a review for a PhD thesis or a master's thesis or for any other project, but it involves a lot more information there. Clinical investigation is one where it's a very well-designed and planned systematic study in human subjects. I think the critical term you have to remember is in human subjects, undertaken to verify the safety and or performance of a specific device. Generally, it involves both safety and performance, but to varying extents. So this is what we mean today by clinical investigation. So commonly, the term clinical trials is also used for this. The term clinical trials is very common in the drugs and vaccines area and popularly used in all the newspapers and so on. But when it comes to the technical areas of medical devices, particularly the regulatory side, now the preferred terminology is clinical investigation. And this is the term that is also specified in our medical device rules of 2017. Medical device rules, instead of safety and performance, they talk about performance or effectiveness. Both terms are used in MDR. They overlap a lot, but there's a small difference. Then the other term the medical device rules uses, which I mentioned, is the clinical performance evaluation. It's specifically intended for in vitro diagnostic studies. So it's a systematic performance of the study of a new in vitro diagnostic medical device on a specimen collected from human participants to assess the performance of the product test, or generally, usually they are diagnostic tests. So kindly remember the differences between these terms, because these are important to make sure that we all understand each other. So one of the important things about terminology is to ensure that you understand what somebody else is trying to tell you and somebody else understands what you're trying to tell them. Now, what is the difference between performance, effectiveness, and efficacy? So, performance is the ability of a medical device to achieve its intended purpose as claimed by the manufacturer. So, if you are a startup, if you're an entrepreneur, you are developing or making a device, tomorrow you're going to manufacture it and sell it. What you put down as its specifications, what you claim the device will do, those become performance. So it includes all your specifications, it includes what you say it will do inside the body or outside the body or how it will be used. So that is performance. Effectiveness, in a sense, is almost same but slightly different. A device is clinically effective when it produces the desired clinical effect intended by the manufacturer. Again, it's what you intend the manufacturer that the device should do, but the clinical effect that it produces, does it cure your disease? Does it solve a problem? Does the heart, you have a pacemaker which is implanted, does the heart, because of the pacemaker's functioning, is the heart functioning as well as it should be, producing adequate cardiac output? So that is the result. That result is clinical effectiveness. There's been another definition which I found slightly varying from this. The device will provide clinically significant results when used as per manufacturer's instructions in the intended patient population. But I think this is the well accepted first one, but this is also used in some of the regulatory authorities. The term efficacy almost means the same thing, but generally is limited to defining effectiveness under an ideal controlled setting. So when you use the device under a controlled settings in a limited population or in a very selected group of people, then the term efficacy is preferred. Now the other terminology is safety. We talk about device safety. Of course, device safety depends on how the user uses it or the customer uses it. And is accompanied by adequate directions and warnings against unsafe use. So assuming that the user uses the device as per your manufacturer's directions and based on valid scientific evidence, the device safety is to ensure that the benefits to health from the use of the device for its intended uses and conditions of use outweigh any risks. So one of the problems in life is that I don't think there's any device which doesn't pose any risks, maybe very small. Some devices can pose high risks. You want to take an X-ray, there is a risk of exposure to X-ray radiation. You want to take an injection, you could get a small infection because the needle or the person taking a blood sample doesn't do it properly. So there's always some risk associated with use of any medical device. Even a small thing like a tongue depressor, for example, if it has a small burr on its edge, it can cut the tongue or lead to a little bit of bleeding. This has been known. And so even very simple devices can pose risks to patients or the users. So safety is to adequately demonstrate the absence of unreasonable risk or illness or injury. So this is one aspect of safety which needs to be demonstrated as part of the clinical investigation. So when do we need a clinical investigation? So a clinical investigation is needed when clinical data is needed to assure or ensure, I think the word better is assure, or demonstrate the safety and performance or effectiveness of the device in human patients or in the intended population. So there is a set of essential requirements which is there in our medical device rules, which have been notified. Essential requirements of medical device were originally published in Europe under the medical device regulations, the directive in 1993. Now it's sort of globally accepted as a very fundamental set of requirements to demonstrate the safety and performance of a device. So the Medical Device Rules 2017 as published by the CDSCO which is the organization which implements the drugs and devices, the Drugs and Cosmetics Act of 1940, under which medical devices are being regulated. It is a department or a unit of the health ministry. So clinical investigations are needed for Class C and Class D devices broadly as per these rules. But again, there are conditions under which you can avoid Class C if you have a good predicate and adequate testing has been done. I won't go into that. But generally, if you have a new device or a substantial change or improvement of an existing device, then clinical investigations will be required in Class C and D devices. There's another international standard called ISO 14155. The latest version is the 2020. It is the clinical investigation of medical devices for human subjects, good clinical practice. So now most regulatory authorities, particularly if you're looking at international certification, will require that any clinical investigation you carry out meets the requirements of the standard, particularly for good clinical practice, or GCP as they're generally called. So the requirements of the standard is it specifies the general requirements like protecting the rights, safety, and well-being of the human subjects or patients who are involved. Ensure that the scientific conduct of the clinical investigation and credibility of the clinical investigation results will be established. That is, you don't do a clinical study which leads to useless results. So basically, the study should be well planned, should have a very strong scientific basis, should be well analyzed to minimize risks, and ensure that at the end of the clinical investigation, you have useful results. It defines the responsibilities of the sponsor, that is the person or the company which really initiates the clinical investigation. Usually the sponsor is also the developer and the manufacturer of the medical device. And then you can have principal investigators. So there are principal clinical investigators, and then there could be other clinical investigators. And the standard also is to assist sponsors, investigators, ethics committees, regulatory authorities, and other bodies involved in the conformity assessment of medical devices. So this is the broad requirements of this standard. And basically, if you follow that, it ensures that your study is well planned. You will be doing a good job, and you will not waste time, effort, and basically place human subjects at risk, which is very unethical. This is the fundamental. So the underlying concept is always that you don't do a study in human subjects which is unethical or which places them at high risk. The standard gives a nice flow chart. So this is the flow chart of the thing. It basically covers the broad. So the sponsor, as I said, is usually the manufacturer or the one who provides the medical devices. And then together with a set of clinical investigators or one clinical investigator, he develops what is called a clinical investigational plan. This is very important. This details how the study will be conducted, how the patients will be recruited, what are the parameters that will be monitored, how long will the patients be followed up for a longer time, what are the tests that will be conducted, how the data will be analyzed, what is the risks involved, how have they been controlled. All that becomes part of the clinical investigational plan. Then the sponsor and the clinical investigators submit the plan to the ethics committee of the institution, of the hospital or the clinical institution where the study is going to be carried out. Now it's the responsibility of the ethics committee to review the plan and ensure that the plan conforms to the Helsinki Declaration on Ethics. So there have been a number of amendments on the original Helsinki Declaration. I think the latest one is in Tokyo. I don't remember the date. So you'll have to check that. So this committee's responsibility to ensure that the plan meets the requirements of the Helsinki Declaration in terms of the ethics and conduct of the study itself. It will also carefully scrutinize how the subjects are going to be selected, How is the informed consent being taken? Is it being taken properly? Are the subjects been adequately informed about the risks involved? And do the subjects have the freedom and option to opt out of the study at any time if required? So those are all the responsibilities of the ethics committee. Generally, the sponsor also appoints a monitor who basically monitors how the study is being conducted by the clinical investigators. Are they recruiting adequate number of subjects to ensure that the study is carried out properly? There's no point saying that, okay, 100 subjects are required, and after 20 subjects, if a clinical investigator turns around and says, no, I'm sorry, I can't find subjects for the study, then remember all the effort and time up to that point becomes a waste. So that is totally unethical and unacceptable. So the monitor ensures that the clinical investigator will do his job in finding the subjects, recruit them, and get them involved in the study. And then the monitor also follows up on all adverse events or adverse device effects. Adverse events may be device related or may not be device related. So he'll keep the sponsor and the ethics committee informed about all these adverse events. It's also the responsibility of the clinical investigator to keep the sponsor and, if necessary, the ethics committee informed about adverse events, particularly if the risks are suddenly high and the adverse events needs to be looked at very seriously. For usually high-risk devices like implants, the monitor may be replaced by what is called a data safety and monitoring committee. It may not be just one person, maybe two or three, generally involving one or two clinical experts. All of you might have heard recently about the vaccine study, the Oxford vaccine study being temporarily stopped when one patient died. And that is generally the responsibility of what's called the Data Safety and Monitoring Board. Basically, they will temporarily hold the study and then look at the adverse event, what happened, because death is a very serious adverse event, and if it is due to the device or the drug, then it needs to be examined very, very carefully to ensure that it is safe to continue the study. So that's what the role of the monitor is there. But of course, it's the role of everybody involved in the study to ensure that, as I said, subjects are not put to undue risk at any point of time. The clinical investigators will collect all the patient data as part of the study and put them into well-designed what are called case record forms or clinical record forms. And so each patient usually will have a set of record forms in which the data is entered in detail. It is very important for the clinical investigator to ensure that the data is recorded properly. I can tell you that from previous experience, I've seen forms which have not been filled up properly. And then when you try to analyze them, it becomes a problem because you don't have the full data on all the patients. And so the clinical investigator will analyze the reports, and usually he'll also take the support of a statistician or others when the final analysis is done and the final report is prepared. So that's broadly how the flowchart of a clinical investigation works. There's a lot of work, there's a lot of money involved in this, and there's a lot of risk involved for patients. We are involving a lot of people, and so this is a very serious business. So coming to the device studies themselves, one thing must remember that device studies are unique. Each device study by itself is different from other devices. So you need to understand that it's a lot more work planning a device study sometimes compared to some of the drug studies. They tend to be smaller than drug trials. We don't rarely will have thousands of patients enrolled. Like if you talk about the COVID vaccine trial, phase 3, they're talking about enrolling anywhere from 25,000 to 30,000 subjects. You won't see those numbers in device trials. Some of the studies may be very novel, particularly the new Me Too devices. But one of the problems in many devices studies are difficult to blind. I don't know if you're all familiar with blinded studies, randomized controlled studies, and so on. I won't go into those definitions. I think I can take them up during question answers if somebody is really interested. Many devices depend on the clinician or physician's technique for effectiveness. Particularly implants are highly technique sensitive, like hip implants or knee implants are technique sensitive. And so if the device is not properly implanted or used by the doctor or the clinician, the results can be negative, or it can even lead to other problems. So this is a challenge. This is one of the reasons that when you plan a study, it's important that the clinical investigators you use are experienced. They know your device well. They're familiar with the device and how to use it. If necessary, they have been properly trained and ensure that they will implant the device or use it properly during the clinical study. If you don't do that, your quality of data can be affected, and it can be a waste of effort, time, and money. Occasionally, device modifications can occur during a clinical trial or clinical investigation. Normally, it's not permitted, but it can occur in the early phase. But it requires you'll have to go through a lot of further documentation and approvals to get that done. But it is potentially possible. This is not permitted in drug trials at all. You can't have a drug and make changes to it during the clinical investigation. Now, the term endpoints to many of you will be new. Endpoints in clinical investigations or clinical trials are the outcome measures that you use to measure the safety and performance of a device. So these are generally called endpoints. They're also called outcome measures. So the endpoint here doesn't mean the end of the clinical study. They are the outcome measures. This is I found it a bit confusing terminology in the initial phase, but it takes some time to get used to it. But kindly remember what endpoints are when it comes to clinical investigations and clinical studies. For medical devices, you will have end up with a diverse set of endpoints. So it can be challenging to define. Many times you need to go back to the recommendations of many international clinical associations who will specify how a particular device or a particular surgical procedure needs to be evaluated. What are the measures to be looked at? What are the testing that needs to be done, or the blood parameters to be looked at, and so on. So device studies, again, the major departure from drug trials are that you generally have only two phases, which is called a pilot study and a pivotal study. I will explain that in more detail. Drug studies, vaccine studies, as you might have recently seen, are usually carried out in three phases. Phase one there is generally what's the purely small safety study, where the drug or vaccine is given to a small set of volunteers, healthy volunteers, usually around 20, 25, less than 30, and then to show that at the sort of estimated dosages, the drug or the biological product vaccine is still safe. Then phase two is when a limited set of safety and effectiveness studies done. It's more a safety study in a larger population. I think now for COVID, I think they are using around 300, 400 patients in phase two. The main emphasis there on ensuring safety and maybe early safety data, like, for vaccines, now they're measuring what is the level of antibodies raised and so on. So the level of antibody that a body generates is one endpoint or one outcome measure. So in devices, we do a pilot study where we primarily look at the safety of the device and some limited data on performance or effectiveness, or you can call it more like efficacy. And then the study moves into usually a multi-center, what's called a pivotal study, where you usually use more than one center. Pilot is done usually in one center and one hospital. Pivotal studies are done in multi-center so that you have more users, more clinicians using your device, and then so that you can demonstrate that when different users are using device, it's still safe. The data shows that it is safe and it performs as you claim it to be. So the overall aim is to support a reasonable assurance of safety and effectiveness for the marketing application. So as I mentioned, clinical investigation of medical devices. So MDR 2017 clearly specifies two phases. First part is called the pilot study. In US, I found the words feasibility study or first-in-man studies also being used in many of the publications from US particularly. I'm not sure what Europeans use. The aim is to establish the basic safety and potential for effectiveness. Generally, the studies are small, usually in the range of 10 to 40 patients. Could be larger, but I have not seen rarely, because remember, all clinical studies are expensive and time is a factor. The longer the time you need to follow up, the larger the cost. So we don't want to make this larger than really needed. Often required prior to a pivotal study to reduce the exposure of a larger group of patients to the risk. So the aim is that you establish a basic safety and you reduce the risk when you move on to a larger pivotal study. That is the primary aim. It is not intended to be the primary support for a marketing application. That is, the data will not be enough to support a marketing application for getting a license or a manufacturing license for sale. Generally, because the numbers are small, it may be difficult to establish a statistical basis for the numbers. So the numbers are generally gone by thumb rules, roughly how many complications you can expect or how many adverse events are likely. So for very serious implants, you may even limit it to 10. But for generally, most studies I've seen vary from about 20 to 30 or up to 40. So the pivotal study usually follows, as I said, the pilot study. Usually the pilot study patients are what we call rolled into the pivotal study. That means the pilot study gets expanded into a pivotal study and it's sort of continued, of course, with all the permissions. Primarily designed to demonstrate reasonable assurance of safety and effectiveness, which justifies benefits against the risks in the use of the device. So the endpoints that, as I said, the endpoints and sample sizes need to be statistically driven. So your number of subjects you want to enroll, you have to justify statistically based on risks. And this is a bit of a challenge. You need a good statistician to generally advise you on how to determine your sample size. Please remember that if you try to enroll a large number of patients, it's going to take longer, it's going to cost you more. While if your patient size is not adequate, you may not then the study may not get approved or accepted for your licensing application. So it's a challenge. Usually defining the sample size, the number of patients to be recruited is always a bit of a challenge and requires a lot of effort and time in the initial planning. So the study should be designed to assess both safety and performance. Kindly remember that. So when you do a risk analysis, when you do a statistical analysis, you need to find out how many patients are required to demonstrate safety, how many are required to demonstrate its performance, and generally the larger of the two is what will define the requirements of the study. Usually the numbers are much larger, as I said, maybe in the range of 100 to 300 patients. It again depends on the type of device, what are the risks involved, what are the complications or adverse events that are likely to occur. One of the problems is if the incidence of adverse events is extremely small, then your number of patients required is much larger to get a statistically significant data on its incidence. That is one of the problems in clinical studies. If your device is very safe, you may need a larger number of patients to demonstrate its safety. It's sort of illogical, but well, the statistics, that's how the statistics generally go to some extent. So the number of patients, you must statistically prove that the primary objective of the study has been established. So the statistics must be good. Generally, you will be required to achieve at least 95% confidence intervals in your statistical analysis. But this can be challenging in many devices. So to some extent, you may get away with putting a confidence level at 80%. I will explain about heart valves where this is what is because otherwise numbers become too large and the study becomes too expensive and it takes too long. Generally, for implantable devices, you may need follow-up durations up to 1 year of the last patient. So your study itself can go on. Usually implantables with 1 year follow-up, I think the whole study will easily take 2 years to complete, maybe a little more. Again, depends on how quickly subjects can be recruited, but studies can be a fairly long duration. These are some of the requirements before the commencement of your study. So you need quite a few documents outlining the basis and justification of the study. One important document is called the Clinical Investigator's Brochure, which contains all preclinical safety and performance data. So if you have done animal studies, all that goes into this, all your lab testing, all that as part of your preclinical safety testing goes into this Clinical Investigator's Brochure, which is shared with both the clinicians, the ethics committees, and even the regulatory authority. Then a clear set of instructions on how the device should be used or applied. And as I said, you need a clinical evaluation report, particularly if you want to comply with the European and US and other developed countries' regulatory requirements. As I mentioned, just to repeat again, the clinical evaluation report is a review of your predicates, of the treatment that is given, all the risks involved, and includes a risk analysis of all your data and shows how you have controlled the risks. Generally, you'll also need to have a risk analysis report or a risk control report as part of your brochure. So then comes the clinical investigational plan, which is, so the statistics is generally based on your analysis as part of your clinical evaluation report. Basically, where you roughly estimate what is the incidence of the various problems, adverse events, and how much numbers are required to demonstrate the effectiveness of your product. You need to attach all biodata of the clinical investigators, showing that you have trained, experienced clinical investigators who can effectively carry out the study. Then a list of institutions taking part. And basically, because if you have to ensure that the site or the institution where the study is done has adequate infrastructure and facilities to carry out the study effectively. Then, of course, before the study starts, you must have the ethics committee approval in writing and the regulatory authorities' approval for the if it is in India, of course, the notified devices are only regulated, and you have to have the DCGI's approval for those notified devices. So some of the prerequisites to meet ethics and regulatory requirements is to address the Helsinki Declaration. This is part of the clinical investigation plan. You have to show that the importance of objectives is in proportion to the inherent risk to the human subjects. This is the fundamental basis of ethics, which says that the risks and benefits are proportionate and the risks are acceptable. You have to carry out a careful assessment of the predictable risks in comparison with foreseeable benefits to the subjects. The risk can be to the users, the clinicians, or to others, So you have to ensure that the benefits are, the risks are predictable and the benefits are known, or the risks have been minimized and the benefits are acceptable. Or the risk-benefit ratio is acceptable, actually, that's the correct term. Then you have also made all efforts to minimize or control all the risks and keep them to the minimum so that the human subjects are not exposed to undue or unwanted risks. So some of the basic elements of submission or risk analysis, as I said, is a very important part today. So what are the potential risks to the patients? What are the adverse events? So how do you find this out? So you look at the current predicate studies or predicate devices or the current gold standard. So for a given treatment, what are the risks involved? What are the adverse events potential? So they could all occur, and you need to try to see what is the likely incidence. Now you have to also see that does your plan mitigate the risks wherever possible. So you plan the study in a way that these risks are mitigated and that the risks outweighed by the potential for benefit. That means the benefits outweigh the risks and the value of the study. So the patient, and then the plan on how your patient will be monitored, what are the follow-up plans, and what are the criteria when a patient will be included in the study or excluded in the study. Because this will be critically looked at. If you exclude many patients who should be included, then your study may not get approved. So you need to judicially design, the clinician has to design his inclusion-exclusion criteria very carefully so that you have a fairly broad population for eventually going to use the study who will, use your device will be included. If your inclusion criteria is very tight, then when you really finally go for a manufacturing license, the regulator may insist that your device is approved only for use in that small set of, the small subset of population. So please remember that the inclusion criteria will also define how your license is provided and the patient group in whom the device will be approved for use. For most drugs and initially even devices, testing in children is normally not carried out. So generally when a device is approved or a new drug is approved, it will not be approved for use in children, particularly below the age of 18 or 16. So if your device particularly targets children, then your inclusion criteria must be very clearly defined that it includes children, because when you include children in studies, then you're getting consent forms and all that becomes more complicated. So all these criteria will affect how you take your informed consent, how much documentation is done. And then, as I said, you have to determine the sample size, the number of investigational centers. You have to justify the number of centers, the number of samples based on statistical analysis. Coming to the documentation, I think briefly on the preclinical data, preclinical evaluation, you have to do a design control and risk management, which is part of your documentation as you do your development. Then the preclinical design verification, including materials qualification, physical, chemical characterization, biocompatibility, all the testing you may do in simulated systems. Like for a hip implant, you may need a hip simulator to show that your hip will really last for 10 years or 15 years. You may need to test as per various applicable standards. One of the common standards, like say for electronic or instrumentation, is IEC 60601 standards for EMI, EMC compatibility. High-risk devices generally will require preclinical testing in large animals, but it can be challenging to do some studies. Like for a hip implant, it's almost impossible to do animal study because the design of the implant, the loading is entirely different from humans. Many spinal implants are also extremely difficult to test in human models. So you may test some sub-features of the device in an animal model, but you cannot completely test a device in many animal models. So there are challenges in designing animal studies for many devices. Then your software has to be validated properly before you get into clinical studies. As I said, EMI, EMC compatibility. Then other thing you need to do is when your quality systems as per ISO 13485 and good manufacturing practices should be implemented for the batch of devices that you're going to make for the clinical studies. Our DCGI or CDSCO calls that a test manufacturing license. So you may have to apply for a test manufacturing license. And at that time, you have to implement the quality systems and implement good manufacturing practices. So if you require a good clean room for making a device for clinical use, you might see these devices must be made in clean rooms. That means the requisite good manufacturing practices should be followed, including environment, clean room, cleaning, sterilization, packaging, everything has to be complete. So in a sense, you need to do a little bit like a pilot production before you can get into a good clinical investigation study. As I said, the clinical evaluation report has to be carried out. This is ideal because it gives you a lot of information on predicates of similar devices tested, collect and analyze the device performance and measures. What are the adverse events? What is their incidence and how are they being mitigated? And based on all this data, you do a risk assessment and identify the residual risks that can be mitigated only by a clinical investigation. So this is broadly what this clinical evaluation report will contain at the end of the study. Now, the other thing you all need to be very clear about is the definition of intended use and indications for use. Please remember that how you define your intended use will define your risk classification of the device. So like a ECG monitor, if you say is intended for use in a clinician's office, it will be class B in India. If you say the ECG monitor is going to be used in ICU or in operation theater, it will become a class C device. So then it may require a clinical investigation. So the definition of intended use is very critical. I think another example is like a scalpel blade. If you say a scalpel blade for normal skin incisions, it is a class B surgical instrument or a class A probably. Well, the minute you say it is for intended for cataract surgery or eye surgery, it becomes a high-risk device. It goes into class D. So you need to define the intended use. This is what you claim on the device labels and the instructions for use as to under what clinical conditions and patients the device will be used for treatment. So indications for use is under what circumstances, under what conditions we would use a medical device. It's a little bit overlap is there between these two terminology. But in your all your literature labels, these are defined as intended use. You have to define the patient population in whom the device will be used, where and when. Broadly, that's roughly what is needed. So please remember again, let me emphasize, intended use will determine the risk class of the device, and the risk classification in turn will determine the type of clinical investigation or clinical study you will carry out. As I mentioned, a class scalpel for general surgical use is a class 1 in Europe or a class A in India. Same scalpel, if you say it's intended for eye surgery, becomes a class 3 in Europe or a class C device in India. The scalpel itself could be exactly the same, except for what you claim as intended use. So please remember there's a lot of difference between class 1 and class 3, or class A and class C. So the sample size for pivotal studies, as I said, you need to balance the need for reliable answers while minimizing number of subjects to risk. This is a challenge. This is where you need a good statistician support to justify your subject size. You don't want your numbers to be large as the cost and time will go up, but you need enough to generate reliable answers. So the number is generally determined by the primary objectives of your study. So the usual methods, just a minute, please. So you should clearly define what are your primary objectives. Like any statistical analysis, you define a null hypothesis and an alternate hypothesis. And then you define the probability of erroneously rejecting the null hypothesis. This is called type 1 error. Generally, it should be 5% or less. That means you have a confidence interval of 95%. But for some devices, it can be challenging to achieve that. So you can justify a larger error if the study requires. Then what is the probability of erroneously accepting the alternate hypothesis? Actually, it should be type... this is erroneously accepting the hypothesis is called type 2 error, and this generally should be 20% or less while taking into account dropouts and other protocol deviations. So the calculation of the size should normally be given in your study plan or protocol. The population selection is based on intended use as defined by you. As a subset, represent the intended population well. You define by age, sex, disease conditions, whether the babies are used, neonatals, pediatric, adults, geriatric, all or some, whether male, female, or both. Disease conditions like severity, is it mild, moderate, or severe, can be combined with other morbid conditions, can be combined with other devices. So these are some of the things you need to address in defining your study population. So objectives, you clearly define under what conditions that they you have to basically your objective defines the main is to verify under usual conditions even that the performance category devices are those intended by the manufacturer. So the study should be conducted in almost the same conditions under which your device will be eventually be used. That's what it means. I'm sorry the language is not very clear there. Determine any undesirable side effects and assess whether these constitute undesirable risks when weighed against the clinical benefits. So if your risks are high, you need to control them. If the risks are acceptable compared to the benefits, then you can justify your study. And then you have to ensure that you obtain firm evidence to support these aims. So as I said, the endpoints or the outcome variables or characteristics that you're going to measure, the endpoints can be measured objectively to determine the benefits and risks. So this is one of the challenges in clinical studies is can all the endpoints be measured objectively? Ideally, they should be quantifiable, but sometimes you may need to make it semi-quantitative as a rating or so on. Occasionally, it may be just a subjective assessment by the clinician. So the endpoints of a clinical investigation or trial are usually included in the study objectives. So please remember that. Then the techniques to avoid bias is one of the problems in many studies, or many times the clinicians themselves who who carry out the study can be biased. Of course, the sponsor is very biased. He wants the device to succeed. He wants the study to succeed. So there's always a bias in clinical investigations. Generally, blinding is carried out. So you can have a single blinding or a double blinding. Single blinding is when the patient doesn't know whether he's getting the study device or he's getting some control or a placebo in the case of drugs. Double blinding is when the clinician himself also doesn't know, apart from the patient, they don't know what they are getting. And in most implants and high-risk devices, this can be very challenging. You can't hide what device is being implanted. The clinician obviously is going to implant it, he knows. Sometimes it's unethical to hide from the patient what device is being given. Like if you look at a heart valve, for example, or a hip implant, you can't tell the patient, you may get the test device or you may get a control device. that becomes unethical because the patient has to understand the risks fully, and it's something that affects him for life. So it's not, so there's a big challenge with devices. So blinding can be troublesome. Blinding can be there in other studies in the therapy, in sort of in vitro diagnostic studies or some of the low risk devices. It's still possible. Then use of controls. So what is a control? In most biological studies, including clinical studies, please remember that there are no absolute measures. There's no way of saying this is the height a high jumper has jumped. So we don't have absolute numbers to say when you will be passed. It's not like an exam where we say, okay, if you get 50% marks, you pass. If you get less than 50%, you fail. And that sort of absolute numbers are not practical here. So most biological studies, including clinical studies, are comparative. The evidence is comparative, and you compare the evidence as better, greater comparison to a control or whatever is the current standard of treatment. So the controls can be your current standard of treatment. It can be another device or a product which is already there. If nothing is there, then of course your current standard of treatment becomes your comparator. Then generally it's also required to reduce bias is to randomize the patients between the test and control. This again can be challenging with some devices. Ideally, randomization is good, but if the cost is going to be very high to have two subjects, you may need to think about it. So we can do non-randomized studies also. So controls can be first thing is the types of controls are concurrent controls. So where you have a predicate device, you have a new ventilator, and you want to test and find out how good it works. So you can have an existing ventilator as a predicate device and then compare your performance against that and show that your device performs equally well or better than the predicate. If you claim your new ventilator has better features, then you have to measure those outcomes and show that those definitely are working on producing a better effect. So the other option is a passive concurrent control where the control group receives an alternative intervention, sometimes no intervention, but is not under the direct care of the same clinical study or investigator. This is very rare in device studies, but some studies it is possible. Crossover controls are where, say, half get one device, half get the other side, and then halfway through you change over the treatment. Many of the established implants, we do historical controls because we already have a large amount of data on existing predicate devices. You could have standards also for these devices. And then what you do is you compare the performance of your new device with the existing historical data, thereby reducing the number of patients who are exposed to in the clinical study for risk. So existing established products like heart valves, hip joints, knee joints, you can manage with historical controls, unless you're having a very novel new product, then you will definitely need some sort of a control. So next, coming to the endpoints or outcome variables, you have to define the relationship between the variable and the treatment. I think that's a critical part. Why you are measuring the variable and how does it demonstrate the outcome of the treatment. So you need to also analyze or look at factors that can affect the endpoint. So many of the factors like age, sex, existing morbidity or health of the patient may influence the outcome variables. So you need to analyze and then your data should show the effect of these variables also. Basically, the variables, the endpoints should provide answers posed by the aims and objectives, should have a direct impact on the claims made for the device. These variables should be directly observable, ideally measurable, objectively determined as far as possible, and subject to minimal bias and error. So directly related to the biological effect of the clinical condition of the patient receiving the device. And eventually, for many of the implants and so on, you want to look at the quality of life. Is the quality of life improved because of the use of your device or your treatment? I'm sorry, I'm taking too long. What's the time now?
5:30, doctor.
Oh, I'm taking too long. Okay. Study site is okay. Pooling data or study site investigators. I think I'll skip these two. I think I'm almost done. Then follow-up reporting safety issues. Maybe we will continue this another day. Statistical analysis is a very critical part. You have to missing values and outliers you need to handle properly because outliers can influence observations and conclusions. They should be very well justified medically as well as statistically. And these are some of the references. I'm sorry, it's been longer than I thought. Sorry.
Thanks. It's okay, doctor. Thank you for the presentation, doctor. So we have a couple of questions in the chat box.
Yeah.
So,
Yes.
Kalarani, do you want to unmute and go ahead with your question?
Can I
Yeah,
close? Can I close the presentation and go back?
Yes, doctor. You can close your presentation, doctor.
hello. Sir, good evening, sir.
Just a minute. Just let me stop the
Yeah,
presentation.
sure.
So that yeah, let me switch on my video so that we can see each other. Okay. Yeah, please go ahead.
Yeah, actually, good evening, sir. I am Professor Kalarani from Tirupati Women's University.
Yeah.
Very nice listening to you, sir. This is the first time I'm getting to know about the clinical investigations of their devices. Actually, I was involved in teaching biopharmaceuticals to the biotechnology students. Where we come across some preclinical trials and all. So I just wanted to know the subtle difference you mentioned about efficiency and efficacy. And you said you consider some of the controlled conditions while assessing the efficacy. What are those controlled conditions, sir, you have to consider to assess the efficacy?
It is not efficiency, it's effectiveness.
Effectiveness, sorry.
The
Yeah, effectiveness.
the two terms generally used with medical devices are performance and effectiveness.
Okay.
Performance is what the manufacturer claims his device will achieve or do, and effectiveness is what really is what the device actually does to the patient or what benefits the patient gets out of using the device. There's a slight difference because effectiveness depends on the clinical outcomes, while performance is what the manufacturer specifies and claims. There is an overlap between the two, but increasingly, originally the European Union relied on performance alone as the manufacturer responsibility, but today they are also moving the world. So when you do a clinical investigation or a clinical trial for a medical device, you're required to demonstrate both the safety and the effectiveness of your device today.
I see. And one more.
Efficacy, as I mentioned, is when you determine the effectiveness in a very subgroup or a smaller population. So like if you test your device only for male patients, for example, for some reason you restrict it to only male patients, then you may claim it that, and then, or you do it in a subgroup, a smaller subgroup for some reason, then the term efficacy is preferred instead of effectiveness. But essentially the parameters are the same. What are the measured parameters?
Yeah, usually in drug testing, sir, in preclinical trials, we manipulate the dosage as per the test species we are considering in the early stages of preclinical trials, and how it goes with, the trials with the devices. Like animal models, how they are considered, will they really consider any animal model before trying on humans?
Generally, yes. If you have a high risk or moderate to high risk devices, and generally it's always recommended that you do an animal study before you get into clinical. Of course, the biocompatibility of the materials used and all that is always done.
Yeah, that's my question, sir.
That anyway is a part of the preclinical. And when we say animal model studies, I'm actually talking about the final device being tested in an animal itself. Like a heart valve, for example, we implanted it in a sheep inside the heart and show that it works. This is the what we call the full, you may call it a full animal model of the actual final device being used in an animal. Now, many devices we can do that, but some devices are difficult to do a full animal model study.
Yes.
Like as I said, hip implants are very difficult to do that because the sizes and the design has to be different. It just is not possible. Hip and knee implants. Spinal implants also are challenging, but nowadays you can do spinal implants in a goat or a sheep model, for example. But it may show the basic performance of the material, but it may not show the wear and tear of the device because the loading is entirely different. Mechanical loading will be entirely different. So there are always limitations to animal studies when you're doing it with devices.
Yeah, exactly. Thank you, sir. Thank you.
Thank you, Kalarani. So the next question is Ram Prasad Srinivas. Ram Prasad Srinivas, do you want to unmute and go ahead with your question?
Hello, sir. This is Ram Prasad. I have a question.
Yeah.
So, I'm working on diagnostics area, which are blood-based diagnostics for pregnant women. So, our diagnostic tests, which are blood-based, come under in vitro diagnostics, and if it is, so what is the regulatory approach for these tests?
Yeah, all in vitro diagnostics are broadly under the group of medical devices definition. But now the certification is slightly done differently. As I said, it's not called our medical device rules 2017 calls it a clinical performance evaluation and not a clinical investigation. I don't know what was the logic for that name, but anyway, that's the title they have. You can check it under one of the sections there. So primarily the study is same. You have a comparator or you have a control. You take samples of pregnant women and then you test it with your kit and with the control kit and show that your device performs as well or even better in terms of sensitivity and specificity.
Okay. So,
does it answer your question?
Yeah, I think so. I think if an agreement analysis between an existing kit and the innovation, or would that be okay?
Yeah, basically it's a comparative analysis. That's why you always have a comparator in all these biological studies. As I said, it's extremely difficult to do a sort of a unique study, particularly in vitro diagnostics, because how do you otherwise establish the like what is 100% sensitivity?
Right.
You take a blood sample, you're testing for pregnancy.
Right.
And how do you know that it's when your say false positives are zero, or what is the actual value of false positives? So you can only do that against some comparator.
Okay.
Unless you have an absolute laboratory method of measuring some enzyme or something which is very well accepted. That becomes a comparator there. So it can be a highly established analysis in a lab. It can even be some sort of a chemical analysis, or sometimes for like say neonatal monitoring we use LCMS methods to measure that particular antigen or antibody or hormone or whatever you're looking for. You understand my point?
I understand.
You can measure a hormone using LCMS, a high-tech method, and then you measure it accurately and then show that your test is as good as that. No problem.
So who should do this validation? Should the
you do it in a good clinical lab, well-established clinical lab. Obviously, the person who actually carries out the investigation should be an expert in this area as a user. Okay. That's why clinical investigations are always also called user validation. Okay. So the user should use it, not you.
Okay, understood. Fantastic. Got it. Thank you, sir. Thank you very much. Yeah.
Thank you, Ram Prasad. So we have a question from Manish Dev. Manish Dev, do you want to unmute and go ahead with your question?
Yeah, sure. Hi, sir.
Yeah.
We're working on respiratory headwear.
I'm sorry, respiratory?
Respiratory headwear.
Okay.
I wanted to ask you what the regulation and certification bodies are for this kind of equipment, and also what's an efficient way to get this kind of equipment to the market? Does tying up with government bodies help, or should we directly approach healthcare sector?
As I said, you need to define your intended use and what is the risk classification of your device. And then whether is there a predicate? Is it something like a generic device of something that's already there in the market? So you need to look at all those factors and then see whether there's a need to carry out a clinical investigation. As of now, offhand, I don't know the risk classification for a device, so I cannot tell you. I mean, tell you, advise you on exactly what needs to be done. I think that would be challenging right now.
Okay, sir.
So look at the predicates. What is the current standard of treatment? What is happening there? What sort of devices are used? What is their risk classification? And how have they been tested, or have any clinical studies been done for them? So this is all part of what I call the clinical evaluation report you need to do. So you go and look at historically what has happened, both the treatment methodology as well as what devices are being used in your case, or in the current treatment setup for that particular disease or for the particular condition. That's why doing a clinical evaluation is a very important part, understanding the clinical aspects of the whole problem.
Okay. Okay, thanks.
Hope that Manisha's question has been answered.
Yeah.
Okay, thank you, Manish. So the next question we have from Akshan. Akshan, do you want to unmute and go ahead with your question?
Yes, I will ask her. Hello, sir. This is Dr. Akshan here.
Yes.
My question is, if the materials that we want to use is already allowed by the regulatory bodies, to be in contact with human skin, then when conducting our clinical trial study and submitting it, do we have to, do a research about those materials and give, certain facts about those materials, or is it okay if we just go ahead with it, sir?
See, all materials, usually you need to have the biocompatibility data for all the materials you use, particularly for devices which are in contact with the patients. And that is part of the preclinical data that you need to submit. So if you're buying already a certified material from a manufacturer who says it's already medical grade and he shares the copies of the certificate, you have to check whether that certificate is acceptable eventually to a regulatory authority. So it's all about risk. So if you don't have enough data, you may end up, then this regulatory authority may end up saying that you have not tested your materials properly before the clinical study. Please remember that your preclinical study should be complete and carried out fully so that your device that you use on patients is proven to be safe preclinically. If you don't do that, then the clinical study may itself investigation may get invalidated totally. So there are a lot of risks in not doing the preclinical testing adequately before you get into clinical. Please always remember that. And the higher the risk of your product, the higher the effort you need to put into preclinical data to show that you don't run into this risk. Because clinical investigations are very expensive. There's a lot of time and effort goes into that. And it becomes unethical if your device is not proven to be safe, from at least with all the adequate data available preclinically. Of course, there's a job of the HS committee and the regulatory authority to review and check that you have done it. But if they miss that, it can lead to other problems for you. So please ensure that your preclinical testing is adequately done, or you have proof from the manufacturer that you're using medical grade materials for the intended application. That's why I keep saying that the definition of your intended use is very critical in defining the amount of data you need to generate both for the preclinical as well as the clinical studies.
Right, sir. Thank you very much, sir.
Thank you, Akshan. So the next question from Anuya Nisal. Anuya Nisal, do you want to unmute and go ahead with your question? Anuya?
Yeah. Am I audible?
Yes, yes.
Yeah. Thank you, Doctor, for an excellent talk. I'm Anya Nisar. I'm a scientist at National Chemical Laboratory in Pune. And we have been working in the area of biomedical implants. So I just wanted to ask you one question. We are in the process of getting our approvals for doing our pilot investigation. So when we design this clinical trial for pivotal especially, is there anything that we keep in mind so that the same data can be submitted, when you want to go to a other market like US, or you're going for other European markets? Is that possible, and is there something that we should keep in mind when we design the clinical trial?
It's always ideal to ensure that you're complying with the international requirements when you do a clinical study. Primarily because clinical studies are very expensive. So doing a well-planned, well-controlled study, I think, is absolute minimum, because by that you also reduce the risk to patients, and by that you're ethically more correct. Because remember, you're dealing with patients. Some of them may be your friends tomorrow, or even your relatives. So it's, I think, ethically very important that we do this clinical studies very carefully, because tomorrow the device may be used in your close relative or a friend or even your children, for all we know. So I would say that yes, it is important. It also ensures that the data you eventually generate enables you to use the data for exporting, for getting a CE mark, or tomorrow you want to export it to US FDA, they may accept the data. But the risks with clinical data getting accepted abroad is usually the acceptance of the patient population in which you carry out the study. Because, let me tell you about the heart valve, which we have done. And I think a couple of years back, TTK got the CE mark for the heart valve, and almost all the data is from India. So one of the questions European technical expert reviewers ask is, how do you show that the data from Indian patients is applicable to European patients? The same question US FDA will also ask. And need to do a lot of analysis to show that the incidence of the disease or the way the disease presents in treatment is similar. And so the adverse events and risks that you may see in Indian population is similar to what you will see in European patients. And so the data, what you generate here in terms of safety and performance, is applicable to European patients. So I did that for TTK for the heart valve. It was a lot of work, but eventually they got the CE mark 2 years ago. So it's a tough thing. So what many, coronary stents, for example, we have, I think, 3 or 4 leading manufacturers of coronary stents in India. To solve this problem, what they did was they generally go and do studies in Spain or in one of the European countries also. So it's a strategy, it's a cost, it's an investment cost, and strategy of how you want to. For wound dressings and that sort of devices, it may not be that difficult to get it accepted. But cardiac implants are very challenging. Cardiac and neuro, you will have serious problems, or even orthopedic, for example, sometimes. Orthopedic and bone plates and screws are easy, but hip and knee, they may turn around and say that the hip designed for Indian patients doesn't suit European patients. It's easy for them to say that. So that's the challenge there. You need to really do an analysis and study the whole thing and see what is best to do. But before doing any clinical investigation, it's good to look at all these issues and then design your study accordingly.
Yeah. Thank you so much. Thanks.
Thank you, Anya. So we have one question from UIC Biotechnology. Do you want to unmute and go ahead with your question, UIC Biotechnology? So I can ask their questions on behalf of them. So okay, go ahead, guys.
Good evening, sir.
Yeah.
I am Bharat Kumar Vairak Innovation Fellow from Anna University.
Okay.
Right now I am doing research in diagnostic kit for urinary strip for albuminuria.
Okay.
Is ethical clearances mandatory for urine sample collection?
Yes.
Okay. Even for a healthy volunteer, is it important?
Healthy volunteer depends on where you're doing the study. See, again, if you're calling it, as I said, a clinical performance evaluation and getting it done in a clinical lab, then you will need some sort of ethics clearance. because you're using patients' personal material, tissues belonging to the person in a study, and then you're going to get an approval or publish it or whatever it is. So all that requires ethical clearance. If you're just doing it in a lab, you may take samples from your own volunteers within your lab and then do it in your college or whatever. That's okay, as long as as taking the sample doesn't cause any risk. While if you want to take a blood sample, there are risks associated. Then again, ethical issues do come in.
Okay,
so like a challenge, you need to be careful. Urine samples are okay. You can obviously collect from volunteers nearby and do that initially. That's not a problem. But blood samples, you have to be a little more careful.
Okay, sir. What is the validation for? What is the validation procedure for this?
That's what as I said, you will have to go back and see what is the current test methods being used, and then use that one of them as a comparator. That's why as I said, look at the history, review what is going on, look at the current methods of testing, what is the gold standard being used for this, and then see, maybe use that as a comparator.
Okay. Oh, one more question, sir. If I am applying for one ethical clearance in one medical ethical council, and can I get sample from other medical college?
No, you can't. See, an ethics committee of a hospital or institute applies only to the patients of that institution. Okay. So if, like say Ramachandra in Chennai ethical committee of Ramachandra in Chennai cannot give the ethical approval for some other hospital somewhere else.
Okay, sir.
Because their job is to protect their own patients, to ensure that their patients are ethically treated. You follow? That's the policy. So the ethics committee of the institute primarily addresses the protection of their own, that particular institution's patients only. Okay. Hello.
I think we lost
one more comment, okay, and hopefully he can hear me. But I think for some studies now, ICMR has a central committee, ethics committee, where you can get ethical clearance for some studies, particularly for institutions that small institutions that don't have their own ethics committee.
Okay, sir. Okay.
It is possible to get it from another overarching institution. It is possible, but it requires you have to talk to them, that ethics committee, and see whether they will review your data for you.
Okay, fine. Thank you, sir. Okay.
Thank you, Varun. So we have the last question from Sai Prasad Poirekar. Sai Prasad, do you want to unmute and go ahead with your question?
Yes, sure. Am I audible?
Yes, Sai Prasad.
Yeah, fine. Good evening, sir. I'm working on a wound healing space, and the device what I'm working on, like a skin spray gun, and spray skin tissues on the burn wounds for rapid. Fine. And currently, I'm doing that preclinical validation with Ramachandra Hospital. And my question is, in the product development stage, when we should do a biocompatibility study, before a preclinical study or after that, like EMC test and all that kind of studies?
EMC, is your device, does it use electronics?
Yeah, there is a use of electronics in there.
Basically, preclinical studies, I mean, all these are part of the preclinical study. So you need to have a strategy depending on cost and when you're ready, depending on where your prototype is. Is it your first final prototype or first prototype? Ideally, for preclinical design verification, all your testing should be done on the final prototype, final working prototype, as we call it. Okay. But sometimes, like if you are looking at a material, for example, you have 2-3 vendors and you want to screen which material is suitable for you, which supplier is more cost effective or something, or gives the correct quality. You may want to do some screening studies in the early phase. Okay, selected studies to show that you have the right quality components or materials. So testing has to be, there has to be a strategy in how you plan your study during your development phase. So because you may need to do some screening of some components or materials or even sub-assemblies, you may want to test all that. But eventually the final biocompatibility, EMI, EMC, all that has to be done in your final working prototype. In fact, you need to submit 3 instruments for EMI-EMC studies under ISO 60601, IEC 60601 actually.
Okay. Okay, sir. And my second question to this is, see, are that non-regulated and regulated device still like there are rules changing every time? So the thing is, currently it is like a non-regulated medical device for me. Okay.
Which one? I'm sorry. This is a wound dressing or what? I mean, exactly what?
So it is a skin dispenser.
Skin dispenser. Okay. So dispensers is something like a wound dressing, is it?
Yes.
Okay. So basically, wound dressings are notified now by CDSCO. You can look at the risk classification. There's a fairly large number of different types of wound dressings there. So you have to go through that list and see how it works. If we are still confused, best is to write to CDSCO today. I mean, you can apply to them and they'll tell you whether it is regulated or not. They'll give, if it's not regulated, they will give you a NOC, a no objection certificate.
Okay, sir. Fine. Thank you.
Thank you, Saitra. So, Doctor, we came to the end. So, before we wrap, I would like to make few announcements. We posted the feedback form link in the chat box. So, I request all the participants to please fill the feedback form for us to improve our upcoming sessions. We are also hosting a Knowledge Tuesday session on coming Tuesday at 5 p.m. on the topic Our Journey in Rising Grants. The speaker is Chilma Parvanta, who is the founder and CEO of Cognosys Private Limited. And thank you, Doctor, for your time and answering all the questions for the participants. Also, the session was insightful through the presentation, Doctor. Once again, thank you, Doctor, for your support and the presentation you have presented, which is difficult but most needed for the startups, Doctor.
So we can have another session maybe a month later if you want.
Thank you, Doctor. Thank you.
Longer session. I mean, we can have more of a question answer session only without any presentation. Also, if there are there's enough interest among the participants to come back and ask more questions.
Yeah. No thank you, Doctor. Then I will arrange that also.
We can consider that if there is enough interest and questions are there. I have no problem.
Okay, Doctor. So usually we will post our session videos on your YouTube channels for the participants who missed out our session. So I hope that is fine with you, Doctor.
No problem. Yeah.
Thank you, Doctor. So thank you for all the participants who joined the session and have a good evening. Thank you, Doctor.
Thank you. So good evening. Have a good day, all of you. Bye.
Bye, Doctor.
Thank you, sir. Thank you. Bye.